Hi, everyone. Welcome to OsteoBites, and thanks so much for joining us today. My name is Christina Iptoma, and I am mom to OsteoAngel Dylan and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Amy LeBlanc from the National Cancer Institute, and she will be discussing her work on new insights into shared molecular and genomic features between humans and dogs with osteosarcoma. And MIB Agents is so proud to support this work with an Outsmarting Osteosarcoma Hero Grant. Thanks so much, Dr. LeBlanc, for joining us on OsteoBites today. And everyone, please feel free to submit questions during the presentation through the Q&A feature in Zoom. It should be in your menu panel at the bottom of your screen, and we will share those with Dr. LeBlanc after her presentation. A little bit more about our guest today. Dr. Amy LeBlanc is a board-certified veterinary oncologist, senior scientist, and the director of the intramural NCI's Comparative Oncology program. And in this position, she conducts preclinical mouse and translational pet dog studies that are designed to inform the drug and imaging agent development path for human cancer patients, specifically those with osteosarcoma. She directly oversees the NCI Comparative Oncology Trials Consortium, or COTC, which provides the infrastructure necessary to connect participating veterinary academic institutions with stakeholders in drug development to execute fit-for-purpose comparative clinical trials in novel therapeutics and imaging agents. Her program provides support to several extramural NCI-funded initiatives, including the Integrated Canine Data Commons and Cancer Moonshot-funded Canine immunotherapeutic clinical trials conducted under the PRECINCT network. We're thrilled to have you on OsteoBites today, Dr. LeBlanc. And before we get started, I just have a couple of announcements. Registration for our Factor 2026 Osteosarcoma Conference. It's going to be June 25th to 27th in Columbus, Ohio this year. Registration is now open. So I'll put a registration link in the chat where you can also find more information. We are also now accepting applications for travel awards, both scientific travel awards as well as Osteo Warrior travel awards. And I'll put a link in the chat where you can find more info and apply. The Scientific Travel Award applications are due March 27th in Proposal Central. And then Osteo Warriors must be nominated by a healthcare professional, and those can be submitted on a rolling basis. And then also just wanted to plug our next Turbo meeting. So Turbo is our tumor review board for osteosarcoma. And our next meeting is next week on Wednesday, March 11th at 5 p.m. Eastern. Clinicians and researchers are invited to join this educational forum to discuss complex cases. And if you're interested in attending or presenting a case, I'll put a link in the chat where you can get more info. And patients and caregivers, you are also welcome to ask your oncologist to present a case on your behalf. So with that, we'll go ahead and get started with our presentation today.
All right, well, let's just jump in then. So this is exciting times for us. We have some great things to share. So from the NCI, I will show you kind of some updates on what we're working on. Of course, some of the Outsmarting Osteosarcoma Hero funded work. And also just, I have some sort of myth, perception, reality discussion points at the end that I thought would be fun for us to work off of for a lively discussion. Okay, so here are some things that we're going to touch on today. I was at a meeting recently where a bunch of veterinarians who take care of canine cancer patients talked about humans being models for dogs, and I kind of loved that. And so we're going to talk a little bit about that in the context of historical perspective, because I think that this group might kind of enjoy hearing about what veterinarians have been doing for the last 40 years while physicians have been working to better the lives of their human patients for the last several decades as well. We're going to flash back to the 80s and talk about MAP and dogs a little bit. One thing that any veterinarian on this call might appreciate is a statement that dogs are not small humans. They're not. Obviously, they are man's best friend, but they have some very specific things about their immune systems that are important. And I think no discussion about osteosarcoma would be complete without touching on dog to human comparisons in the immune system realm. We're going to talk about tools and some of the new toolbox offerings that are available to researchers in the community. And then we'll also touch on genomes, transcriptomes, and AI. Oh my. So I think just historically, it's important for the community to know that veterinarians have been thinking and working and talking and researching osteosarcoma for a very long time. So you can go back to the mid 1940s and find the first histologic descriptions, and then a really nice sort of first big description of the clinical management and diagnosis of osteosarcoma came out of Penn's veterinary school. 194 dogs in 1969. And this was from Robert Brody, who's sort of thought of as one of the fathers of veterinary surgery. And what you see from the literature is as you move forward into the 70s, 80s, and 90s, that veterinarians were trying a lot of different things to manage canine osteosarcoma patients because it is such a common veterinary problem. So they're the first descriptions of therapeutic strategies that mirror a lot of what was happening in humans at the same time go all the way back to amputation and immunotherapy using BCG treatments from the 70s into more modern chemotherapeutic approaches. The first report of methotrexate with leucovorin rescue was published in 1978. Then we move into like the platinum and anthracycline era of the 80s and early 90s. Along that same time, there were some interesting surgical and other local therapy techniques, radiopharmaceutical development happening in terms of like limb sparing and metastectomy approaches, and samarium-based radiopharmaceuticals were all described in the like sort of late 80s, early 90s. And here are just some fun screen grabs of these papers. And I just show you this because you can go back pretty far into the 50s, 60s, and see where, veterinarians were really trying to describe this disease and make sense of it. And then also paying very close attention to what was happening in humans to sort of better the management of their patients. And I have this picture of a letter written from Bill Hardy from Sloan Kettering back in 1973 saying we should create a veterinary cancer society. And that society was actually founded formally in 1976. So if anybody's wondering, we've been working on veterinary cancer for quite a while, and we have been using, I think, humans as models for dogs to try to develop our own sort of strategies for patient care. A couple of interesting things when we talk about the comparative aspects of dogs as a translational bridge between mice and humans. Again, some really early studies, both in dogs and humans, occurring about the same time. So 1966, 1967 for humans, risk of osteosarcoma development was linked to body size. So the bigger you are as a dog and maybe the taller you are as a human, there was a recognition of a risk that was specifically higher in those populations. We also, I think, jointly started to wonder about certain things that could be prognostic, like serum alkaline phosphatase levels, the location of the tumor, and then the percent necrosis after neoadjuvant therapy. So these sort of facets of clinical management started to, sort of show up in the literature as things that people were thinking about. And then the LMTPPE, rafamotide story sort of showed up in the mid 1980s. And you see several waves of papers come through the late 80s and early 90s where veterinarians were working very closely with Dr. Kleinerman and her team and others within the community to try to understand whether or not dogs could aid in the development of that compound, but also sort of immunotherapeutic approaches writ large. And then also just characterization of the disease was happening at that time too. And so this was before like really a lot of modern techniques that we use now, like sequencing, were even sort of possible at outside of very specific research laboratories, or technology hadn't even really been developed at this point to do some of these things. But you can go back and see that there are descriptions in the veterinary literature about PET scans being performed on dogs in the 80s as FDG and modern pet radiopharmaceuticals were coming into the clinic, development of cell lines and xenograft models. And then you can definitely appreciate the uptick in research and data as genomic profiling kind of came into its own with next generation sequencing techniques in the early 2000s, 2010s, and definitely into the present day. And so this is just a screen grab of the size sort of comparison. So here you see the JNCI published in 1966 that canine bone sarcoma risk as a function of body size, and really shortly thereafter, the landmark Joseph Fraumeni paper in Cancer said stature and malignant tumors of bone in childhood and adolescence are inherently linked. One important thing to just point out here too, because my goal for today is to show you what we're learning, but also to sort of compare and contrast what looks similar and what's not similar between dogs and humans, because it's always an important thing for us to recognize again that dogs are not small humans and humans aren't dogs. One thing that's like sort of an interesting thing to meditate on is the age at diagnosis. So for me as a veterinarian, this is an old dog disease. This is a disease that typically affects dogs in their 8th, 9th year of life. And that's usually where you see that peak incidence in the dotted line on this graph. There is a very small but significant population of dogs who develop their disease kind of young, like puppyhood, early adulthood. But typically, they're older. Which is quite different than the pediatric AYA space, where the peak incidence is certainly within childhood or early adolescence or early adulthood. So I always sort of wonder about that and what it might be telling us or not telling us. And I think it's an important thing for us to think about if maybe the community agrees that this is a disease of developing bone and maturing bone and a differentiating bone. And so we have to think about what the age of onset might mean or not mean in the context of dogs as patient models.
So for dogs who do develop disease earlier in their puppyhood and younger age, are there any characteristics like certain breeds or anything in common?
Yeah, so they're usually really big, the big dogs, the giant breed dogs that like the wolfhound, like the really like mastiff, like the dogs that really are like going to top out at 130, 140, 150 pounds. But having said that, like I've seen osteosarcoma in a mixed breed dog that was, 8 months old. So, I think that's one of those things where it falls to the veterinary community to sort of think carefully about how we report our caseload and even the data sets that I have that I've shared and still continue to work on, we can look at their ages and say, maybe there's a distinct group of features that we can assign to dogs that are less than 5 years of age, maybe, or less than 2 years of age. It's a relatively small population, but it is worthy of further consideration. And I would say just also, people ask this, question about skeletal maturity, because it is really an important facet of human bone development and osteosarcoma development as well. And the canine skeleton generally reaches full maturity, depending on the breed, between 6 and 12 months of age. And so the bigger dogs probably don't reach full skeletal maturity until they're 10, 11, 12, maybe even 14 months old. Smaller dogs are going to reach maturity more quickly, but smaller dogs are generally not the ones that get osteosarcoma. So there's just a lot of really interesting bone development questions in there that I think are worthy of further consideration. And this is a slide that actually I got from Sean Khanna, who many of you probably know, and one that he used a lot in his presentations. And I think I show this to you just because it, again, just gives you the historical perspective of what veterinarians have been trying to do from a cancer standpoint. So a lot of what's on here is not just osteosarcoma, but other solid tumors too, like the things that veterinarians have developed and worked on for melanoma and for drug development activities, and some of the landmark things that our program here at NCI has done under Sean's leadership, where, some really important things were established that helped create a framework for us to have a research network that was not just clinically focused, but reached more into the research realm and, created some nice inroads with the pediatric and AYA research communities on the physician side, so we could talk and share data and learn from each other. And I took a nod from this and made sort of a next sort of generation timeline of this. This is very focused on osteosarcoma for the purposes of this talk. But I feel like in the last 10 to 12 years, we've kind of, started to move more in a sophisticated way, as of course the tools and funding have, in an interest, really a sort of groundswell of interest has developed, where, we've gotten some sophisticated projects here that you can see, where we've gotten good clinical trial infrastructure in place. We've gotten really good federal funding that didn't really exist not, much long before 2014. And a lot of really important immunotherapeutic things have been established and networks for collaboration have been established. So I feel like, we continue to sort of grow as a community and really try to keep pace with what's happening on the physician side as best we can. And again, just for the purposes of this talk, and for those who may not be as familiar with comparative oncology, this is a bit of information that I learned from a colleague of mine, where we talk about canine and human together as patients, and then we talk about the rodent models that we're heavily relying on in the cancer research community. And when you think about the advantages of studying dogs and how much more closely they resemble their human counterparts, these are some points that are really important to, I think, consider. Dogs and humans are studied across a wide range of age, whereas the rodent models we use are inherently young. They're baby mice. They're 4 to 6 weeks old. Not many rodent studies take rodents into the aged, over a year of age. Unfortunately, canine and human patients and people in general are increasingly overweight, whereas the mice we study are lean and perfect and young. And they live in, these mice live in a specific pathogen-free environment. They have a homogeneous genetic background, and we experimentally induce tumors that arise quickly, are noted quickly, and treated quickly. And you really can't rely too heavily on mice for tolerability or toxicity signals. Whereas all those things are really the opposite on the canine human side. We share a home environment. Dogs get vaccines just like humans get vaccines. We share a lot of our immune education and immune editing and epitope spreading in response to infectious agents in the community and in the environment, very outbred genomics. Tumors are going to co-evolve and be subject to immunologic pressure. And dogs and humans have a lot of similarities in terms of how they respond to therapy, both good and bad. So, especially within the immunotherapeutic realm, you can really capture adverse events pretty accurately in dogs. They have the same sort of repertoire of adverse events, and we have good metrics that are based off the CTCAE guidelines in humans to capture and characterize those. And I think specific to osteosarcoma, again, as I mentioned, this is a really common veterinary tumor with really wide community awareness and interest. We see this disease affect both pure breed and outbred dog populations equally. More and more, and as I hope I captured in my sort of mini history lesson, veterinarians have tried really hard to adopt human strategies for chemotherapy, surgery, radiotherapy, immunotherapy, and we've really tried to adopt good staging and characterization criteria to capture events and other sort of facets of clinical trial and tumor management. One thing that's pretty important to note is that dogs don't really have a standard of care that they have to adhere to. So certainly when I am in front of a family with a dog that has osteosarcoma, we provide them lots of options to choose from. They don't have to do anything, obviously, but they can choose to say, I don't think I want to give my dog chemotherapy. I think I'd like to do something else. And that's a nice opportunity for us to sort of evaluate new treatments in the context of a patient that hasn't been heavily pretreated with other things. And then just a reminder about workflow. So we'll come back to this when we talk about MAP and other sort of adoption of therapy across species. We've sort of figured out after decades of trying that the most effective way to manage local disease in dogs is through a full limb amputation. And we typically do that up front to, again, get the tumor burden down and control pain and discomfort. There are certainly situations where limb sparing or other limb-on management techniques like stereotactic radiotherapy or palliative radiation with chemotherapy. And there's some really interesting newer techniques that are being evaluated to sort of let a dog keep its leg, but also have its tumor be treated. So we're working on those as well, because maybe a pet owner says, I don't want to amputate my dog's leg. And that's okay too. We can think about ways to approach a dog without having to do that. But the nice thing about doing it is that we get the entire end block specimen with adjacent normal tissue and draining lymph nodes and all kinds of things that really help advance research questions. And that workflow is quite different than what happens in people. So I'm going to run you through some scientific themes, and then we're going to end with some sort of myth, perception, reality, which I hope will spark some questions from the audience and some good discussion for everyone. So we'll start with tumor microenvironmental profiling that has strong translational relevance for humans. And some of you may have seen this slide before, but I'm going to run you through it again because it really does form the basis for how we're starting to think about the disease in dogs and how we can develop methods for comparing and contrasting to humans. So this is work that was done by predominantly a very talented postdoc in the artificial intelligence resource in my branch, Sushant Patkar. And he worked with my staff scientist, Jessica Beck, who's a veterinary pathologist, to create a method for assessing the tumor microenvironment of canine osteosarcoma and relating it to humans. And this slide shows you bulk RNA-seq data from 198 untreated canine primary tumors in the middle, and then nanostring data, mostly from metastatic lesions that were collected at necropsy. Those are in green, and some primaries that were also subjected to nanostring profiling because they didn't have good enough RNA. And so a couple things that I just want to point out before I go any further. It's really hard to get really good RNA out of bone tumors. It's a difficult tissue to work with. Our sequencing facilities have very high bars for quality assurance and quality control. So for us to do RNA-seq in this context, we really have to pass pretty high bars. So a lot of our tumor tissue just didn't pass QA/QC, which is why we only had 198 of the over 400 in our collection pass muster here. Nanostring profiling is another technique has a dog-specific reagent and toolbox sort of platform where you get an 800 gene panel. So it's still expression data, but it's a down-selected group of genes. And it's a lot more forgiving for doing gene expression profiling off RNA that's been somewhat degraded. And so a lot of these nanostring profiles came from FFPE. So it's just a way of complementing and augmenting the data set. But what this shows you is when you can take bulk RNA-seq data and do computational methods that deconvolute the transcripts into estimated abundances of cell types that represent those genes. And what you wind up with here are 3 distinct subtypes. So there's an immune-enriched subtype at the top that has a lot of enrichment of genes that point to immune cells like T cells and B cells and NK cells. And then you also have an immune-depleted group, the ID group at the bottom, that really, there's not a whole lot going on there in terms of immune enrichment. And then you have this really interesting middle group that has representation of immune cells, but also has genes that point to extracellular matrix, stromal components, and endothelial cells. And so that becomes important later as I talk about multiplex imaging and immune cell exclusion, which is one of the things that we are starting to appreciate through spatial profiling techniques in humans. And then eventually we're working on that in dogs as well. I think the other thing to take away from this slide is that you can see these 3 TME subtypes that are distinct in terms of their functional and genomic features, not just in the primary tumors, but you can also see it in the metastases that are matched to these primaries. So this technique of TME subtyping really has value because you sort of want to understand and appreciate that just the context of the primary tumor may not indicate what that patient's metastases will look like. And there can be differences across geographic locations. So the nanostring data comes from, lung, liver, kidney, and a whole bunch of other places like bone or soft tissue or brain or eye. So the postmortem exam in dogs really helps us generate a data set like this so we can look across not just the lung as a site of metastatic disease. So this is great, but it gets better when you think, okay, well, does this have value for people? And in fact, it really does. So on the bottom left, you see a Kaplan-Meier curve of disease-free interval in dogs. And what you see here is that the immune-enriched subtypes of primary tumors, those dogs do better with therapy than dogs that have an IE, ECM, or an immune-depleted subtype. And when you take this same algorithm that's been trained on dogs and you apply it to people, it also predicts outcome in people. And so those same 3 TME subtypes are conserved across species and predictive of outcome in both dogs and people. So using publicly available data sets that were very specifically selected to only interrogate the transcriptional profiles of primary tumors from patients that did not have metastatic disease at diagnosis, because that's a really important thing to make sure of for an apples to apples comparison as much as possible. The immune enriched subtypes in humans do better too. And I guess maybe that makes sense, right? So there's a greater immune awareness of the tumor. And then the immune the TME subtype designation also predicted metastatic disease developing within 5 years of diagnosis. So this is, a nice first step in the bulk RNA-seq realm. The next thing that everybody's kind of doing now is spatial profiling. So, the bulk RNA-seq is like you throw all the fruit in the blender and you hit blend and it turns into a smoothie. But spatial gives you sort of like the fruit tart, right? So I can't remember who does this analogy. It might be Rosie Kaplan, who I love, where you see where the strawberries and the blueberries and the pineapple are arranged. Well, you want to get that spatial context because it really helps you know what cells are talking to each other and what cells might not be talking to each other. So we applied the genomics, the NanoString genomics spatial profiling tool to a tumor microarray from our outcome-linked biobank. And this small experiment actually gave us a lot of interesting insight in that. And here we got differentially expressed genes that separated the dogs into two groups. So we had a group that did pretty poorly, that dogs developed metastatic disease very quickly despite therapy, and then a group that did much better. And what distinguished those groups of dogs from each other, among many DEGs that are sort of shown here in this volcano plot, but also the representation of through cell deconvolution of immune cells. So the dogs that did better had more CD8 positive T cells and macrophages than the dogs that didn't. So where we're going to take this next is to take the NanoString genomics spatial profiling tool into a larger group of canine primary tumors that we have matched normal bone and matched metastases from them. So we can look at like non-tumor osteoblasts and look to see what genes are expressed there as opposed to the primary tumor tissue, as opposed to the metastatic tissue from the same patient. And we also have data that is not ready to show quite yet, looking at spatial profiling of tertiary lymphoid structures that form around primary tumors in dogs and compare the expression profiles of those sort of lymphoid aggregates to the draining lymph node that's been collected at the amputation surgery. Because what we're wondering is that there's different sort of immunological repertoires between the draining lymph node in the, say, inguinal or axilla area of the dog and the lymphoid aggregates that are forming directly around the tumor tissue. And we know that the tertiary lymphoid structure presence predicts a better outcome as well. So again, it points to immune recognition of tumor tissue being a good thing.
Dr. LeBlanc, can you just review that volcano plot?
Yes. So what you're seeing here are differentially expressed genes, and there are the dots are different colors because they denote the statistical significance. So everything that is gray is not interesting. Things that are orange, genes that are associated with an orange dot, are sort of interesting, and the things that are significantly interesting are in light blue. And I would point out, again, when you look to the left, those are genes that are enriched in the good doers. So it's a little this is set up sort of maybe differently than other volcano plots that you've seen, but those that are to the left are good. When they're enriched, that's good. And so there's a couple of really, obvious genes that are like P10. So you kind of want to have P10 around. It's a good tumor suppressor gene. And that's highly associated with a good outcome when it's there. And then you've got ones that are associated with a low DFI. And so there's some collagen genes in there. And again, and matrix genes. So you kind of wonder about this immune exclusion thing. So if tumors are upregulating a program that sort of pushes immune cells physically away from the tumor mass, or at least keeps them from getting where they need to go to present antigen to antigen presenting cells and then elucidate a further, immune response, that's not great either. So I will say that this is genomics. It's a nanostring, and so it's only 800 genes. So that's the caveat here that, there are better and other sort of spatial tools like, Visium HD has a canine probe set that we're going to play around with from the 10X group. And there are other sort of opportunities, I think, for looking at the whole transcriptome in a spatial context. So we're kind of investigating what might work best in the tumor tissues that we have. And I know other groups that I'm going to show you here in a minute are going to do that too. All right, so let's talk about multi-omic profiling. And this is where we shout you guys out for helping us work on this. So multi-omic profiling is where you take a group of tumors and you say, well, I want to look at them from a couple of different facets. I want to look at whole genome sequencing to determine mutations, copy number, structural alterations, those kinds of things. I want to look at their gene expression profile. And then I want to look at other sort of modifying events that might might impact gene dose and gene expression relationships. And for us, that's DNA methylation. So that's where methyl groups sort of control how much of the message gets through. If you're playing a game of telephone, so methylation is like the mute button. So what we did here, and I have 55 plus because we started with 55 dogs, and now we've got, because of your support, we have funding to look at much more than this. So we have funding to create multi-omic sets around more than 100 dogs of primary tumor, and then more than 100 matched metastases, mostly from the lung. That's where we'll focus initially, to create these sort of data sets that really help enhance the framework we have in hand. And the data here really just helps you get at some interesting questions about biology. So what's modifying gene dose and gene expression relationships? And then they give you a nice sort of pipeline of things to mine in terms of druggable targets and new therapeutic opportunities, and really nice opportunities for deployable biomarkers for patient stratification. Because I think, one of the things that I've heard from our community that really resonates with me is that this is a very heterogeneous disease, yet the treatments that we typically prescribe don't reflect that heterogeneity. So we need to, on the dog side, I think we can make some inroads into identifying patient subtypes and then having better clinical trial designs that reflect that heterogeneity. So this is just a couple of examples of this work. So here I'm showing you whole genome sort of global methylation patterns across a group of canine primary tumors. And this work through Anjali Garg and my group, who's the computational guru, essentially shows us that there are two groups of dogs. There's a relatively hypermethylated, globally hypermethylated group, and a hypomethylated group. And the reason that's important is because all the outcome data associated with these tumors tells us that if you're hypermethylated, you're probably going to do better than the median's outcome for standard of care, versus hypomethylated, have a poorer outcome. And then she went further to relate the cluster assignment from methylation to the TME, the 3 TME groups that I talked about a minute ago. And so it turns out that if you're assigned to cluster 2, meaning you're relatively hypomethylated, you're also more likely to be immune depleted. And so that's an interesting observation. I'm not saying it's causative, but it's an association where, we're trying to reconstruct the series of events that led to a tumor. The ways that we can do that are make these observations and associations, and then go back to try to figure out what came first, what came second, what came third. So this is just one example of how you can take multi-omic data to get at some of those questions. And this is newer data that has to do with MYC. So MYC is a big topic in terms of potentially a prognostic biomarker in humans. And Anjali was able to show on the left, you've got whole genome sequencing data. In the middle, we've got our methylation cluster assignment, whether you're MYC amplified or MYC not amplified, and then some bulk RNA-seq data from the same tumors that gets at the types of cells that are present in relative proportion across MYC amplified and MYC non-amplified tumors. So let's start from the left. What you're seeing here is a Kaplan-Meier curve on the top that shows two groups of dogs. There's a blue curve that's MYC amplified, and those dogs don't do as well as if you aren't MYC amplified. So that's a new finding. We never really had the data to say maybe MYC has prognostic value in dogs too. And when you are MYC amplified, you are also more likely to be immune depleted. And this data and this sort of observation in the primary tumor context is very similar to data that's coming out of Dan Regan's group looking at metastatic disease and the contributions of MYC and an immunosuppressed environment. So we were happy to see this sort of association come out of our primary tumor data, because it could be that it's a program that is active very early in the disease process itself. Maybe this is an association or a series of events that occurs very early in disease progression and is carried forward into the metastatic setting. So that's great that we can put our heads together there and see that our data sort of converges. When you look at the context of methylation, so if you're MYC amplified, you're more likely to be hypomethylated, which was already, we knew, a bad sign. And now we've got MYC amplification, sort of also compounding the poor outcome of those dogs. So, again, not causal, but an important statistical association that's worthy of further follow-up. And then when we take that into the expression or RNA-seq realm, you see that through cell deconvolution, if you're MYC amplified, you don't have as much immune cells in your tumor, which go along with this immune-depleted TME subtype. So we're really interested in what this might mean in terms of deployable biomarkers for dogs, because I think that for us to be successful in putting good clinical trials together, we need to sort of be able to tell our patients apart in terms of the types of disease they have and assign them to rational therapies that are matched to their patient subtype and molecular and genomic features. Okay, now we're going to talk about AI a little bit. And so again, Sushant and the Turk Bay lab here in the artificial intelligence resource at the end in the intramural program worked again with Jess, my pathologist, to ask some questions about whole slide images and AI-based feature extraction. So this is where you take a bunch of H&E images of tumors and you create a method for feature extraction. And in this case, the features are many, but the obvious sort of way to train an algorithm like this is to tell it, well, here are the chondroblastic regions of the tumor, here are the osteoblastic or giant cell rich or areas of hemorrhage or necrosis. If you annotate those areas and then you teach or train an AI algorithm to detect them, and then you tell it, show me if these patterns have any value in terms of predicting outcome, because we know what happened to every dog in this set. Could that be a predictive algorithm or prognostic sort of factor for canine osteosarcoma? And then maybe it could be transferable to humans, because histologically it's kind of impossible to tell them apart. And so this paper from a couple of years ago says, yes, you can actually do that with this cohort of over 300 dogs. They sort of clustered in these ways based on the histologic patterns that were recognized. And then when clusters 1 and 2 were combined and compared to cluster 3, that was significantly different on the principal component analysis. It turns out that cluster assignment actually predicted outcome as well as patient weight. And so this is where you get it, the bigger dogs do worse. Heavier, bigger dogs maybe have a worse outcome than smaller dogs. And that's a factor that's sort of come and gone in the veterinary literature over time. Sometimes it pops out as significant in a Cox proportional hazards analysis like this. Sometimes it doesn't. In this case, it did. And so what we're doing now with this algorithm is taking it a little further. So Sushant and the other members of the AI team here have developed a tool called HistoTME. And so HistoTME is a solid tumor assessment of H&E images that, through the magic of AI, assigns features that have to do with molecular signatures that are inferred from histologic patterns. And those molecular signatures are outlined in the boxes there. They have pro-tumor, anti-tumor, malignant cell-associated features, angiogenesis or stroma. And this really meshes nicely with the idea that canine and human osteosarcomas have these subtypes that we associate with immune enrichment or extracellular matrix enrichment or not. And so what Sushant did with the first batch of patients that we obtained from COG, actually, through a data use agreement with COG. We had a first pass over some of the patients from the AOST 0331 trial. And this is just a first pass. So we're continuing to apply HistoTME in this context to see if we can get these curves to separate and have the feature extraction algorithm tell us that if it's able to actually just off an H&E image predict outcome. So we have over 700 images from COG trials. And so this is a developing story that hopefully I can update you on later this year. Okay, finally, we'll get into some spatial biology. And so this is where we've been working with canine tumor tissue, normal tissue actually, from canine cancer patients that undergo a postmortem exam. So I mentioned earlier that this is a pretty common thing for veterinary patients, and it really helps us understand what might be happening in the normal tissue of dogs that have cancer that may or may not develop metastatic disease. So not every dog develops in this context, I'm going to talk mostly about lung, but also liver and kidney. So not every dog with osteosarcoma develops lung metastases, and we don't really understand the factors that control that. And so having access to normal adjacent tissue is really important and an exciting opportunity. So what we did here, and again, I'll just remind you, because the data from this study really, again, call back to this 3 subtype TME foundational finding in the bulk RNA-seq context. What we did is we took 2 groups of patients, canine patients. We had lung tissue from a dog that had lung metastasis, and then lung tissue from a dog that didn't develop lung metastasis. It may have had metastasis in other areas, but it didn't occur in the lung or in the liver or in the kidney. And so the question is there something that we can detect in the gene expression of this normal lung? So histologically, it looks normal, but we know it's not. We know that there are changes that occur at the microscopic level that support metastatic outgrowth or may suppress metastatic outgrowth in these areas. And so this was the first sort of study that tried to address that. So these are just a snapshot of the findings. And this paper literally just got accepted like yesterday. So it's in press, and hopefully you'll see it out soon in the open access forum. But the key finding here was also really gratifying to see that it is linked to some ongoing work, again, from the Colorado State group with losartan as a CCL2 inhibitor. So when you look at, this is normal tissue. So this is not tumor tissue at all. This is geographically distinct normal tissue from lung, liver, or kidney from these 2 groups of dogs. Either, yes, I had metastatic disease, or no, I didn't. But they all had osteosarcoma. You see that there's an overlap in CCL2, CXCL10, and some ECM genes that belong to that collagen superfamily. So all 3 of these geographically distinct organs are upregulating these genes to support metastatic outgrowth. And it's a consistent finding. And when you take a signature like this and you overlap it with human NAT studies, which are pretty few and far between. Most of them have to do with radiation injury and looking at adjacent injury of brain and lung in human cancer patients. And you can find some of this through TCGA. There's 100 gene overlap between human and dog there. There's no human osteosarcoma data for us to compare to right now. So what I can do is show you that this is what happens in dogs, and that the yellow and sort of brown bars show you, again, through cell deconvolution, what cell types are up in the, yes, I have a metastatic lesion. So there's fibroblasts and some monocytic lineage cells, and those are appreciated across lung, liver, and kidney. So it's really interesting to find that. And again, inherently validates the use of losartan as a CCL2 inhibitor in terms of metastatic disease, which is an active area of research both within dogs and in humans. And it really is strongly linked to this notion that there's an extracellular matrix signature that might have an immune exclusionary purpose, a pro-tumor purpose that could be targetable. So stay tuned, because I think this is an area that we're really interested in generating more data using spatial profiling techniques and looking across species. And to do that, one thing that we did was develop a nice panel of antibodies that cross-react across mouse, dog, and human. This is another paper that really, I think, provides good information to the community. So if you're interested in trying to do cross-species multiplex imaging and you want one antibody that works across all 3 species, we figured out what that antibody is. And we know what clone and what manufacturer and what dilution and all the sort of technical stuff. And we did that for these cells that are outlined here. And then it's just a pretty multiplex picture to show you what it looks like when you use the Opal 6 platform to image it. So this is another sort of just tool that helps us visualize where these cells are and how they're relating to each other. In this case, this is metastatic disease. So, I mean, I think what we're trying to do and the way that we're thinking about it is to characterize heavily these facets of canine biology. We want to integrate and compare and contrast them to what's available in people. We all want new druggable targets and new therapeutic strategies. And there are ways that you can bring those back to tractable and informative mouse models, or bring them directly into dogs as new pipeline clinical trial options. And then we just keep generating new data that helps perpetuate this ongoing cycle of discovery and advancement. Okay, so finally, this is the fun part of this talk that I'm hoping to get some chat about it. Okay, so these are some things that I've just heard over the years, and I don't know how universally sort of held these beliefs are, but I like the idea of dispelling myths. So we're going to tackle some of these today. So myth number one, dogs with osteo are not treated as aggressively as humans. The reality of that is that's actually partially true. Okay, so I showed you where veterinarians back in the 80s and 90s were trying to give things like methotrexate with leucovorin rescue. We were trying to give high dose dox and platinum drugs together. We tried and tried and tried through the 80s, 90s, and early 2000s to do this. And it was about 15 years ago that we sort of said, okay, we're really not doing anything but making dogs really sick. And we're not really moving the envelope too much here. So I think as a community, and I'd love to hear other people on the call that would like to have an alternate viewpoint, I think we've kind of figured out that single agent platinum is sort of where we live right now. And we typically use carboplatin because of nephrotoxicity concerns. And main issues around canine treatment have to do with quality of life and costs of care. So a lot of veterinary, pet owners, most pet owners maybe don't have funds to just endlessly handle hospitalization and toxicity, and they really want their dog to be at home with them. So I think you can turn this into an advantage, though, because if there's no real standard of care, it gives you opportunities to study new approaches and new techniques. So that's myth number one. Myth number two, dogs' immune systems are very different than humans, so they are not informative models for immunotherapy. This might be partially true, but I think very context dependent. And I'm going to show you some really recent data from the community that gets at this issue. So it is true, dogs are not people, people are not dogs, and they do have similarities in the ways that their immune systems function and the way that their immune systems are represented in the cell populations that are grown and born out of the bone marrow. I will tell you that there are clinical studies that show definitively that dogs that develop cancer and are aged, they're in their 6th, 7th, 8th year of life. As they age, they have similar alterations in their innate and adaptive immunity as human cancer patients. And when you give them chemo or you radiate them, you impact their immune systems. We have known this clinically for a long time. What's new, though, and really exciting, is that there are now single-cell transcriptomic atlases of the canine bone marrow and of immune cell subsets that allow us to really do some nice functional comparisons. And this work pretty much definitively shows that canine T cells are a little different. They produce a little less interferon gamma when they're similarly stimulated. Macrophages appear a little bit more responsive to activation by interferon gamma than human. And canine granulocytes, which are neutrophils, mainly express CD4, which is sort of weird. So you just need to know that. And I think if you're interested, please look at these papers because they are beautiful, beautiful examples. One of them is a really lovely preprint of a bone marrow atlas that is coming out of the Colorado State Group. And there's just really rich, nice data here now to show this is what's really happening. And I think very context dependent. So anytime you're thinking about immunotherapy and you really want to understand how it might work in dogs and how that can be related to humans, I think really carefully ask yourself, what cell types am I expecting to stimulate or suppress? Do I have the tools to really understand whether or not a dog could be informative in this context, and not sort of paint it with a wide brush to say, nope, it's not helpful. Okay, myth number 3. There aren't sufficient research tools to compare canine and human osteosarcoma. That is partially true, but I think getting better. And I think I pointed out a couple times in this talk about where there's some good, new computational tools, methods. I would say that even the data availability and published tools and data sets have just really overwhelmingly improved in the last year alone. It's really great to see the work coming through to benefit everybody. And it can be difficult to find cross-reactive antibodies for certain assays like Western blot or IHC. There are a couple of companies that have come through with some canine-friendly tools. NanoString provides, the canine IO and genomics profiling tools. You can spike in gene options if what is in the panel isn't sufficient for you. So there are things that are getting better, but this is certainly an area for improvement. Let's see, myth number 4. Okay. Canine osteo does not share meaningful molecular alterations. This is false. And I think that, hopefully our group as a whole can work together to drive this home. Transcriptomic signatures and histologic findings really help point in the direction of biologic convergence. The single cell sequencing data sets are beautiful from the groups that are generating them. They indicate highly that we've got conserved ecosystems in both primary and metastatic lesions. And I really feel like there's a lot of evolutionary sort of convergent mechanisms. So maybe the upstream events might be different, but what comes out in the wash is the same. And that's probably why histologically and clinically in response to therapy, you see so many similarities. And there's just a couple of, this is hard to see, but you don't really have to sort of read it too closely to understand. This is a beautiful preprint of an integrated single cell and spatial transcriptomic analysis across species and sites of disease from Ryan Roberts' group and many collaborators from the community. And the sort of 4 dot plot panels that you see from top to bottom, there's human patients at the top, human PDXs right below that, dogs right below that, and then underneath that are mouse models. And so you can really appreciate how similar the dogs are to the human PDXs and the human patient samples. Just by looking at this visually, it just makes sense. Okay, finally, trial findings in dogs are not informative. I would say that this probably really depends on your point of view and the question that you're asking. Again, maybe I'll close with this thought that if you have a very heterogeneous disease, you need to do some patient stratification to help hunt down the therapies that are going to be meaningful for trials that are addressing certain types of therapy. Human studies, I think, need more correlative biology for us to be able to make some comparisons to what we're doing in dogs to what's possible in people. Immunotherapy, as I mentioned before, really needs to be studied in comparable patient subsets and TME subtypes to be informative. And I will just say that, in the dog world, dog owners are really excited about participating in trials that have new treatment options available to them. And I think from a veterinary perspective, I'd love to see us really emphasize progression-free survival rather than overall survival, because that's the meaningful connection between dogs and humans. We want to see better progression-free survival. In dogs, because we have euthanasia sort of complicating that issue, we really need to focus on that as a progression-free survival as a clinical trial endpoint so we can get the most meaningful translation between dog and human. I'm going to stop there. I've left a few minutes for questions. I need to hugely thank all the people in my group and our alumni and other groups that we work with, both within the NCI, NIH, and beyond. Thank you, MIB agents, for all of your generous support and cheerleading for us. Really appreciate it. Happy to take any questions.
Thank you so much, Dr. LeBlanc, for that great overview. And everyone, please feel free to add any questions for Dr. LeBlanc in the Q&A, and or if you just have comments or reactions to some of her Mythbusters. It'd be also great to see. Maybe the question, kind of stemming off, I guess, your last myth about clinical trials, and also maybe a joint question kind of related back to how aggressively dogs are treated versus humans. So you reminded me that MTPPE had been studied in canines and then trialed in humans. And though not FDA approved today, it is still used somewhat commonly for osteosarcoma patients, certainly in other parts of the world outside the US. And then the Listeria vaccine trial also was like a nice example of study in dogs that then moved successfully to human trial. And so curious, like because, and especially one of the last slides that you showed, right, looking at the different, the TME and the spatial analysis, showing the similarities, dogs and humans, not so much mice, this has always been a question of like, can we better use canines as a way to move things to trial? Because, we all know often sometimes something might work in a mouse, but then just we just don't see that happening in humans. And so it seems like there might be that step in between that might be a better indication of how successful it might be in humans, which would be the dog. But what makes a study or something a good candidate for moving into a canine study before?
Yeah. And I think there are a lot of things to consider. I mean, I think it always comes back to sort of, if there's credentialed target biology between dogs and people, that you're really confident that that target is expressed, that you have a biomarker, you have a means of assaying on target effects, and that you're confident that you have enough dogs in the study population to see an effect. And I think I'm going to bring up the listeria for a moment, because I really feel like that was a trial that taught us a lot about immune fitness and not so much about HER2. And I think other people in the community probably also recognize that the dogs that did really well in that trial, it was not overall a positive study in the dog realm, but the dogs that did better clearly demonstrated their ability to mount an immune response. And I think that's the key. I think that that's maybe the lesson from that story, and maybe also from murfamurtide, where it's a therapy that will work as long as you have the ingredients available to you to respond to it. And maybe what I'd like to see more of is if we can identify these subpopulations up front, we can do more rationally designed trials. So, we're really trying to enrich for a subpopulation so that we can have more of a precision medicine approach. I recognize that's going to be really a lot easier for me to do with dogs than it would ever be for people, but I still think that there are inroads to be made there. And it all starts with trying to get a better idea of how to characterize and identify patient subsets up front. Instead of looking back and realizing, oh, well, if we had known that this dog had, I don't know, a MYC amplification or a PTEN deletion or, some other genomic feature or TME subtype, I think the advent of liquid biopsy techniques might help us get there. Because if you have a surrogate that you can repeatedly sample in the peripheral blood to identify responsive versus non-responsive cell populations, that would preclude a lot of complicated things like multiple tumor biopsies and other invasive procedures that, nobody wants to have to go through.
Right. So if I'm understanding correctly, it's a little bit of both. I mean, I understand for clinical trials today in humans, because it can be so difficult to enroll participants, that versus you're just trying to get as many patients as you can, and then doing the crowd studies that you mentioned that we need more of to better understand who's responding. But you're saying also there's some advantage to kind of having some basic understanding of who is the best candidate.
Yeah. And I think it would be great to see more correlative biology as a part of human trials. And I know it's hard. I know it's hard to get all these samples and to keep track of everything and to, match it back to individual patients. And some of these things are just a lot easier to do in dogs because we have so many patients. And when you don't have as many patients, everything just gets harder. So if there's ways that we can provide insight there, I think that's what we want to do.
Great. Okay, so we have some questions that came in that I definitely want to get to. So first, are there different subtypes of osteosarcoma seen in pure breed versus outbreed? This is also unique to canine populations. How do these populations help inform research and translation to humans?
So I think this is one of those questions that always comes up about the breed-specific alterations. And I think, so here's my 2 cents about it. I'm not an evolutionary biologist. I don't really study dog breeds as dog breeds, and as, I don't have the depth of research understanding there. But what I'll tell you is I think they're the best opportunity we have to look at predisposition syndromes because they have such a strong evolutionary grouping. And there are great ways to identify predisposition syndromes that might already be recognized in human populations. You can draw parallels to those in dogs. And then you can also look for therapeutic vulnerabilities. So one thing that we're working on is, and it pops into my head when you bring up breed, because there are some breeds of dogs that are known to potentially have a higher incidence of osteosarcoma, like the greyhound, because of their CDKN2AB loss. And so there's some GWAS studies help identify some of these things, where if you have really big groups of related dogs and pedigree analyses, and you have case control, and you can say, what was it about the germline of this population that might have predisposed them to get osteo in the first place? Well, sometimes those germline mutations or losses, rather, are associated with other genes that modify the tumor microenvironment. And so by doing those studies, then you think, okay, so maybe a segment of chromosome 26 that's lost, or chromosome 11 in the dog, because remember they have 38 pairs of chromosomes, you can identify a germline loss, or that might be a predisposition that might be sitting next to genes that also have a pro-tumorigenic effect. So that's how I'm thinking about it. I don't know that in the veterinary space, we have labels to put on breeds of dogs that say, oh, the Rottweiler osteo is this versus the Greyhound Rottweiler osteo looks like this. I don't think we can do that. And I think it might increase the predisposition of those breeds to get the disease, but I don't know that it has any therapeutic implications. Hopefully that answered your question.
Great, thank you. Okay, next question. Considering we are therapeutically treating the metastatic osteotumors, how do TME subtype profiles compare between the matching primary and metastatic tumors from the same patient?
Yeah, that's a great question, and one that I didn't have a chance to get into. But I can tell you this is our data set, so it's not, it needs to be validated in other hands, in other groups of dogs. But when we did the TME profiling of the primary, and then we lined it up next to the mets from that same dog, there's no correlation. So you can have an immune-enriched primary tumor and have immune-depleted metastases, and vice versa. Now, that's just our data set. I think other groups have found that there's more conservation across the single cell sequencing space, where you have representation of certain cell types that are present in an ecosystem of the primary tumor, and that same ecosystem is conserved in the matched metastatic space. So, at least in our data set, we didn't find that. But I think you can. This is just the signal that we need to look more, right? We need to have more consensus. And those are also very different assays. Single cell sequencing is very different than bulk RNA-seq cell deconvolution. So it may be possible for us to recognize, and I recognize this openly, that bulk RNA-seq doesn't take into account the spatial context and is not nearly as sensitive as a single cell technique. So it's possible that when we dig into the relationship between a primary and its matched metastatic lesion from a spatial context, we're going to find something different. And I think the other thing I want to always remind myself of is, when we're thinking about therapy for dogs or for humans, typically that's therapy that is taking place in the context of all the local disease being done. It's gone. It's out of there, right? What we don't know is whether or not the microenvironment of that primary tumor set up the distant sites to have the same or similar microenvironment, meaning if you had an immune-enriched primary, did that somehow influence the nascent niche of metastatic disease that's already been set up in the lung or the liver or the bone or somewhere else? Those are the relationships that we need to dig into deeper. And that's where I think liquid biopsy might be really, really helpful. So I'm looking forward to that field developing more in that context.
Great, thanks. I also just want to say I loved the smoothie fruit tart. I'm not sure enjoy them in the same way that I have been.
I borrowed that from Rosie Kaplan. So Rosie, if you're out there, thank you.
Okay. I know we're at the top of the hour, but we have some great questions that come in. I want to make sure we get to those. So next question, have you identified any particular effective ways to collaborate with radiation oncology in canine models? The technical details of treatment are similar to humans, but the logistics and access to care can be more difficult in canine patients.
Yeah, that's a great question. I was actually just at the radiation research meeting, winter workshop on comparative oncology, and sort of talking with radiation oncology about the things that they're trying to do, and the realization that dogs as cancer patients could really participate there. There's some really interesting things that can be done with radiotherapy techniques for bone tumors, because more and more, I think dogs provide an opportunity to think about new ways to keep the limb on. Not every dog is a good candidate for a limb amputation if they have concurrent orthopedic disease, or they're just not a good candidate, or their owners just don't want to do that. So I think, our program here has a clinical trial consortium, and we have been in existence for over 20 years, and we've never done a radiotherapy study, and it's probably time to change that. So I look forward to actually entertaining ideas that incorporate radiotherapy into new treatment strategies, and I would love the idea to participate in that more, because I think there's lots of opportunity.
Thank you. Okay, next question. How do the logistics of canine clinical trials differ from human trials? And what aspects of osteosarcoma canine trials are more conducive to enrollment and follow-up, and what are the barriers?
So, I mean, I think the easy things are like, this is just a really common disease. So I think barriers to enrollment are pretty low. Most pet owners will want to hear all the options. And I think the barrier often is geography. So if a clinical trial is only being conducted at a handful of sites across the country, that can be really hard. One of the things that I'm hoping to actually address through that is rethinking the structure of our trial network. Because right now you saw maybe on the map that there's only 19 sites, and they're in the US and Canada, and they're pretty spread out and far apart. I think there are ways to be more responsive to different types of veterinary practices that see these patients, not just at veterinary schools, but also at other referral centers or even general practices. So those are things that I'd like to work on. And, you'd be surprised, logistically, it's pretty similar to a human trial. So we have informed consent paperwork and case reporting forms and, red cap databases that capture data, that keep track of samples, that have, visits spread out for the owners to come back for, procedures and sampling time points and all these kinds of things. It's actually our way. I think we modeled the way that we do trials off the way that people do human trials, because we felt like we needed to be, sort of integrated that way.
Right, thank you. And then we'll close it out with this last question. This is from Amanda Marinoff, who I'm so glad is here, because I was actually going to reference some of her great work, kind of building consensus on biomarkers for humans. And so she asked, you showed beautiful data demonstrating multiple methods for risk stratifying canine osteosarcoma. It's gratifying to see the seeming overlap with findings in humans. How do you think about putting all these diverse stratification methods together? Do you think MYC amplification is sufficient, or do we need a combined model integrating genomic findings with methylation, TME profile, H&E findings, et cetera?
Yeah, so that's an excellent question. And you kind of pulled out maybe the next few years of work here that I think it's a recipe. I think that one of the things that's going to be really important for us on the dog side is to make sure it's something that's deployable clinically. So IHC is one of those things that lends itself well to these things. And I didn't get into it in this talk, but in the paper that that I showed those 3 TME subtypes, we actually backed up that cell deconvolution data with IHC. So we have a panel of IHC markers that serves as a surrogate for that TME signature. So technically, you could assign a TME subtype based on an IHC profile that, working with veterinary pathologists, we could come up with like an algorithm for grading the different intensity and proportion of cells that stain to say this particular IHC readout is an IEECM ID tumor. Moving into MYC, we're working on ways to interrogate MYC in the canine tissue space. I would love it to be, again, a deployable assay. So something like MYC IHC is something we're looking at. We're looking at MYC ddPCR between fresh frozen and FFPE tissues to make sure that we get good ddPCR readouts with assays we're working with Bio-Rad on that. And then we're working on fish as well to try to understand whether we can use fish probes for canine MYC and other genes that appear to have popped out of that analysis, like P10, to see if we can identify biomarkers. And then the third layer of that is the AI. So I think more and more we have already seen this. I think the community has seen this, that AI is becoming a part and parcel of pathology now. Digital pathology and whole slide images lend themselves really well to AI techniques of feature extraction and prognostication. So maybe someday I will be able to show you sort of the 3-pronged approach to canine patient stratification that is a recipe of genomic findings, TME profiling, and AI-based feature extraction. And it can be all wrapped up together in a way, like a rubric maybe of assigning risk and promoting sort of certain druggable targets for certain populations of patients. That's sort of what I dream about being able to do. And so, this is the yeoman's work of trying to get there. So that's a good question. Thank you.
Yeah, no, that's great. And certainly there seems like there's lots of opportunities for that work to inform all the biomarker work and risk stratification work in humans. So thank you so much, Dr. LeBlanc, for joining us on OsteoBites today and for making it better for osteosarcoma patients. More information on this and all OsteoBites can be found on our MIB Agents website at mibagents.org, on the MIB Agents YouTube channel, and at your favorite podcast place. We hope you can join us next week on March 12th. We are going to be joined by Dr. Afrin Petawala from UCSF at the Sweet Cordero Lab, and she's going to discuss her outsmarting osteosarcoma-funded research on identifying mechanisms of chemoresistance in metastatic osteosarcoma using molecular barcoding. You can find our OsteoBites lineup for the next few months on our website, and you can subscribe to our YouTube channel or follow us on Spotify so that you never miss an episode. And if you have ideas for future topics that you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again so much, Dr. LeBlanc, for spending time with us today. And thanks to all of you for taking time out of your day to join us. We hope you can join us next week on March 12th when we chat with Dr. Afrin Petawala. Thanks, everybody. Thanks so much, Dr. LeBlanc.
Thank you. Pleasure. Take care.
All right. Thanks. Bye.
Bye.